In this video, I'm going to fill in some of the gaps that were left out in the previous videos, explaining the details of the visual system. So here I have a rod and a cone, and I'm just going to draw out our pathway very quickly. Remember that a rod carries black and white information and a cone carries colour information. The information from the cones gets sent to the bipolar cells, which then sends the information to the ganglion cells. The ganglion cells then send their axons out of the eye, and that becomes the optic nerve. And the same happens for rods. So here's the optic nerve here. Now the strange thing about the eye is its organization. This is actually the back of the eye, which means that light comes in from the front of the eye through all these apparatuses and then to the photoreceptors. And where the optic nerve dives through the back of the eye, that's your blind spot. So the fibres from this side are the fibres from the rods, and the fibres from this side are the fibres from the cones. So these fibres travel to the LGN. And on a closer look, the LGN looks kind of like this. It has these layers. And these layers are where the cell bodies lie. It has 6 of them, and we name them 1 through 6. And 1 and 2 are slightly different. I'll get to that in a moment. Fibres from rods synapse on layers 1 or 2. The ones from the ipsilateral eye, or the eye from the same side, synapse on layer 2. Fibres from cones can synapse on the other 4. Ones from the ipsilateral side synapse onto layers 3 and 5. So if this is our left eye, these fibres would be coming down to our left LGN. Now the fibres from the other eye would cross through the optic chiasm and here they are here. And the fibres from the rods of the contralateral eye synapse onto 1, or layer 1. And the fibres from the cones synapse onto layers 4 and 6. And this is the input to the LGN. Now these 2 types of layers have 2 different names. These ones are parvocellular, and these ones are magnocellular. Parvo means small, and magno means large. These are because under a microscope, cells in layer 1 and 2 are large, and the cells in 3 to 6 are small. So these fibres continue on, and from the LGN, they travel to the occipital cortex, or the occipital lobe, which is at the back of the brain. And this, of course, will be the left occipital cortex. And these fibres travel down individually, so they haven't mixed yet, and they travel down and synapse in the occipital cortex like this. This is a huge amount of fibres in the brain, because vision is a very complicated sense. And these can be seen as a big white matter tract known as the optic radiation. That's all I want to talk about here, but remember that the visual system is very complicated, and there are even further details that we could discuss here. We'll talk about these in later videos.